Okay, great. So I have a couple of disclaimers that I need to put out up front. Number one is I'm probably going to be the least interesting of the three presenters. I'm a chemist by training and not a pharmacologist. So take that for what it's worth. The second is I won't, owing to the nature of how CARB-X works, which is to fund and support projects. We don't do projects on our own. I won't be able to share case studies and data with you from programs, obviously. And so therefore, what I thought I would do is talk through the requirements at different stages of development that CARB-X has for therapeutics, with a specific focus on the in vivo efficacy, and then relatedly, as a program matures, the in vivo manifestation of secondary pharmacology and toxicology. So with that, I'm going to get underway, and I'm going to try to keep us on time. Okay, so let's see, how's this work? Okay, I thought I'd start by just reminding you that we do have an active funding call, and I'll come to the timeline at the end of this presentation for those of you who might have an interesting program that you'd like to submit. But I'm showing this slide to highlight that of the 4 themes that we elevated as eligible for funding from CARB-X this year, there is a focus on direct-acting small molecule antibiotics with a focus on serious infections caused primarily by gram-negative organisms. It doesn't sound the least bit new to you, because I think this has been a theme all along. But to say that this continues to be a challenge. If you look, just last week, Valeria Gigante at WHO released the updated clinical and preclinical pipelines. And while certainly there's a lot of activity, it's pretty diffuse, and we still don't have a broad spectrum agent for these serious gram-negative infections, particularly those that have the options of both an intravenous and oral form. So just to say that's a focus here. And I say it also because then underpinning the success of a preclinical program must be the demonstration of efficacy and relevant models of infection. So I thought I would start by sharing what we view as typical activities in a hit to lead program, which is where the earliest stage that CARB-X indeed will fund. And so, I won't read this word for word for you, but essentially at the hit to lead stage, we want to see that there's some reason to believe that optimization of a hit is possible. And usually that would come in the form of some preliminary structure activity relationships, an understanding of the chemical scaffold that delivered the hit and what could be harvested from that, and some general attributes of activity, particularly against some sort of target. Ideally, it'd be great if you had structural activity, meaning crystal structures or the like. But we understand not all targets are amenable to that. And then some sense of cytotoxicity in that sort of early read of whether there's any reason to believe that there's a window. And that the stuff can be made. So that's kind of the base level. And then importantly, to demonstrate in vitro some activity against wild type representatives of the pathogens in the target product profile set. So for instance, if it's hospital-acquired bacterial pneumonia, and it's in an IV form, we're going to want to make sure you have some baseline Pseudomonas activity, not activity with a fully permeabilized and devoid of efflux strain pumps. Again, doesn't have to be great, but just a direction of travel that gives you a reason to believe. But importantly, and specific to this webinar, at the end of the stage, so you don't have to come in with data showing efficacy, but at the end of the stage, that there is some data to again give encouragement that the lead series that would be developed in the next stage does act in some relevant animal model of infection. versus an untreated control group. And I'm going to talk a little bit more about that in the upcoming slides. And then otherwise, again, we don't require at this stage that you have a whole secondary farm package or even pharmacokinetics at this stage. We prefer the sort of cheap and cheerful, does it work scenario, and I'll highlight that also in the next slide. And then importantly, some attributes that suggest that the physical chemical properties of the HIT or HIT series can be developed appropriately. So if the intravenous form is the desired form, then some sense that aqueous solubility is achievable is important. And if it's oral, either PK that is suggestive of optimization for a credible oral form, or some CaCO2 cell permeability data that highlights both influx and efflux, again, to demonstrate that oral bioavailability is possible. So just some comments, and this is really driven by stuff that we see, which frankly drives us crazy, is that, you know, in most cases, and certainly not all models are amenable to this, but in most cases, neutropenia is desired. And again, that's really for the purpose of showing that the compound works and can work on its own. Likewise, the model, and I believe Jen certainly will talk a lot more about this, and again, not my area, but you know, you want to show that the starting inoculate, that the bug actually takes off before you start doing treatment. Otherwise, again, it's hard to sort of deconvolute what the compound is or is not doing. Another thing to underscore, and the bold indicates the degree to which this drives me crazy, but models really should have a positive control, which, by the way, isn't necessarily the comparator antibiotic, but this is something that the model was developed on, that you know is going to work, and work, you know, hopefully, ideally, the same every time. So they have a benchmark that, okay, it's if the compound didn't work, it's not because the model didn't work, right? So it's really important, and it seems so obvious, but it's often not done. If a comparator is used, it should be used in a way that's been reported in the literature. Otherwise, it's really not a fair fight. And that the dosing regimen should be reasonable. And at this stage, there's this tension because people, you know, there's a desire, I get it, I used to do this, to show that the compound works. And some people will go to Herculean efforts, including dosing every hour or every 2 hours for the duration of the, you know, 24 hours of treatment. That seems a little bit excessive to us. And then finally, you know, just some comments on the model. Certainly, I think we all know that the neutropenic thigh model has become the gold standard for early screening. We know that it works. We know what to do with the data, and in many ways has taken the mantle from the peritonitis model, which was established, I think, in the 30s. But then it sort of goes down from there. And so if people are developing a product for different indications, you know, we want to make sure that we're not putting too high a hurdle or too much of an emphasis on a model that may not have been standardized or well validated. And so I say that because, and I don't know what Jennifer's going to talk about, but together with Combine and ICON and Eurofins and Hartford Hospital, you know, we are undertaking an endeavor to aid in the standardization and back translation of lung models of infection in mice as a way of making sure that we all as a community understand what the results mean and how to use them, and how they are likely to predict clinical outcomes. And so I won't have time, but love to, you know, share that at some point, the work that we've been doing, and you know, it's starting to be published and talked about in meetings. But then it really goes down from there. So urinary tract models of infection seem like a great idea. But given the number of clinical failures we've had recently, despite the fact that they work extremely well in UTI models of infection, tells us we have some more work to do there. And gonorrhea models are new. So 5 years ago, maybe it was a little more than 5 years ago, when the company where I was developing a gonorrhea product, there were no animal models. And so you know, how do you know how to judge what's going to happen? Now there are a couple, and I think they're converging. and hopefully we'll have more back translations that feel comfortable utilizing them moving forward. Okay, other comments. I am now off time, so I'm going to go quickly here. But other comments about efficacy at this stage, at the hit the lead stage, is that we don't expect that the model is incorporating the latest and greatest clinical isolate with resistance phenotypes and genotypes well defined. Rather, we just want to see that it's a wild type strain that's well validated, again, with a good control molecule to understand that the molecule has some basis for efficacy that can be optimized in the next stage. And then importantly, and I think this is a controversial topic, but we prefer for people to do an MTD and go straight into efficacy rather than to do PK, because at this stage it's much more efficient. So you have to have a good bioanalytical method if you want to do PK, and that's also costly. And so unless there's some reason to believe the molecule is behaving poorly, we prefer to see tolerability and efficacy, and then to follow up later with pharmacokinetics that would lead the way to PK/PD later in the program. I wasn't asked to do this, but I do want to put this right on the table, and hopefully we'll have time to talk about this in the open session. But everything that I've said is true for small molecule direct acting antibiotics and maybe large molecule direct acting antibiotics too. But there's a lot of activity in the non-traditional space, including in the CARB-X portfolio. And this is an area where I think we don't know how well the animal models translates into the clinic. When the programs don't work, the easiest thing for the product developer is to say, well, of course, why would it work? These models were trained and developed on small molecule direct-acting antibiotics. Maybe that's true. Maybe it's not true. I think some work needs to be done there. Now, furthermore, many of these non-traditionals are going to be used as add-on to standard of care or adjunctive to standard of care. And so how do we best assess that preclinically? Often what we see are people, for instance, will use some substandard dose of meropenem plus the compound. Is it the right substandard? How do you know? How do you judge success? And so these are things that we struggle with every day, and at least presently our portfolio is weighted very heavily towards non-traditionals, and so having a sense of what to do there will be very important. Okay. Then I'm just going to go on. I'm not going to read this slide to you, but let's focus on the bold here. So in the lead optimization stage, now having set the table for activity and efficacy, this is where we like to see more in-depth experiments. And so early PK/PD analyses being done to assess the driver. Also, of course, more strains are embraced. Ideally, that would span the range of in vitro activity that you see, so that not everything is in your, way susceptible range, but a range that's necessary. Recognizing, of course, that all isolates don't grow well in these animal models, and so some practicality has to come in there as well. And then, of course, this is where you're going to want to start building out your safety farm and your toxicology package. And these are very important because these are the yin and the yang. So your efficacy is one part of the TI equation and your toxicity is another. And so doing those early non-GLP toxicology studies, which don't incorporate a single animal of each species, and that's great. And people can always say, well, it's a benchmark, but it's one. And when things don't go well, of course, the one is always the outlier. So design your toxicology experiments as well as you design your in vivo efficacy experiments. And then at the IND enabling stage, of course, it's just more of the same. So build it, build it, build it. More in the animal model world. Again, maybe more models of infection, but certainly, again, more strains, more even range of potential doses, et cetera, to really sort of focus in on what's your human likely dose, and therefore how you're going to set yourself up in going to the clinic. And then in the first in human stage, it doesn't stop there. Everybody knows this, I think, but just to say that alongside of gathering exposure and safety data in humans, you want to continue to refine your idea of what efficacy means so that when you go to your proof of concept, you're choosing a good dose to go in and maybe bat around that to demonstrate efficacy in a small, phase 2 program. So I think that's it. I'm not going, because I am over time, talk about our work in the lung model, but happy to do so at another time. But I would be remiss, since CARB-X does have an open funding call, if I didn't say that the first cycle is well on its way. I see some names of people who were in that cycle Bravo, and we are now about to bring those programs to the advisory board stage. But I want you to watch for the 23rd of September when our portal will open for funding call 2. It will have a base of everything that is in the first funding call, but especially in therapeutics, there's going to be a little add-on. So watch out for that, and please do apply. So with that, I'd like to thank you. I'm looking forward to the next presentations and the conversation.
Yeah, thank you so much, Erin. Yeah, maybe I forgot to introduce you, but I always assume that people know you so as the Chief R&D of CARB-X, but sorry that I didn't do that in the beginning. So, but I'm always assuming the people. So, good. So I think what we will do, we'll just take one, maximum 2 questions now, and then the other question we'll take in the panel discussion. So, Jon from SSI, you have a question. Maybe you can just say it loudly. Yeah, thanks. Go ahead.
Thanks, Fred. No, so Erin, great talk, thanks. So you mentioned the 24-hour thigh infection, neutropenic thigh infection, as a go-to for hit to lead proof of concept. I was wondering, 24 hours, if we say it's Q4 dosing, if you're in Europe, you might also need to have people there during the night to look after the mice. So already at this stage, that could be quite an effort because of the 24 hours. Would you ever consider shorter duration models, single dose, 2 dose, 8 hours of duration?
Again, at that stage, I don't know that it's going to be Q4, but I mean, often we see people do, at the 2 hour, maybe 8 hour, and then a chaser before the end. I think that the beauty of the standardized neutropenic model is just that people know how to do with it. And so, again, everything is conversation, but for us, that just lends enough credibility to feel comfortable moving forward.
Okay, thanks.
All right, I don't see any urgent questions. So what we will do first, as I said, we're going to move first to the second presentation, and then Erin, if you can stay online then for the panel discussion. Okay, but thanks a lot for this presentation. Huh?
Thank you. Sure.
Good. Francesca, while I'm taking your slides, can you also introduce yourself so that I don't forget it again to introduce people?
So thanks for letting me this duty. So hello everyone, and thanks Frederik for inviting me again to contribute to this panel discussion. I am Francesca Bernardini. I am VP in vitro biology at Evotec and the IMR therapeutic area lead. I am also a chemist by education. However, I left chemistry a while ago, and because I discovered that microbiology and antimicrobial new antibiotics discovery was much more interesting. So I've been doing that the last 20 years. And I will present a showcase in my presentation that is somehow the contribution of quite a few people present in the audience today, so that we recognize their work as well. So
just give me a second.
Yeah.
Can you see my screen?
Yeah, not full screen.
Yeah, let me just put it on full screen. Now is better. Is it okay now?
It's not full screen, so I don't know.
Let me try again. Sometimes it helps just to go. Okay, let me just put it back on full screen. Now?
Not yet.
What about you, Tolga? How do you see?
Yes, if you could go to window and then go full screen.
Yeah, that's what I. Yeah. Okay. So
we just see the whole file with the all pages. on the
Okay.
left side.
Yeah, I know. Let me just try again. So share and then otherwise I can make it a little bit bigger. Okay, full screen. What about
Yes,
now?
perfect.
Yeah,
Ah,
please.
okay. I didn't do anything different, but just sometimes it helps just to do it again and again. All right, here you go, Francesca.
So if you could please move to the slide number 3 after the disclaimer. I guess that quite a few people present today were already there at the first presentation, so I will very quickly go through the first 2 slides, which are a simple reminder why you need a TPP to drive drug discovery in the antimicrobial resistance space. And so basically, this would pave the path for designing the project strategy and the associated screening cascade, with clear criteria for progression associated to the screening cascade that will be used as checkpoint at each stage of the project to assess what is the chance of success to achieve what is the final goal of the project, and of course de-risk it as early as possible. As Erin mentioned, we're facing more and more the let's say the application of new modalities to address antimicrobial resistance, and for this particular kind of approaches, to have a TPP is to me quite critical because it will allow to combine in vitro and vivo data, but also including the multidimensional, multidisciplinary approach that needs to be taken, also to address critical aspects that may have not been explored before for this kind of modalities. I'm talking in particular about the MPK, so including, of course, efficacy models, the CMC that could be somehow a challenge for new modalities, and of course, safety, as for some approaches may not be, let's say, the comparators may not have been tested before. So let's say the scenario still needs to be set up. And in terms of disease models or efficacy models, the TPP may also be a guidance to set up a dedicated model if you want to represent a specific pathological condition associated to infectious disease. I'm talking about, for example, using specific clinical isolates, like in the case of cystic fibrosis models, for example, or this could be used to qualify models on the basis of host infection biomarkers if host-directed therapy is envisaged. So overall, the TPP can be used to perform a gap analysis during the whole drug discovery process and ensure that the project has the maximum chances of success before moving to the next step. If you could please move to the next slide. This is one example of a target TPP of a generic candidate which may have the potential to address multiple indications. For example, here as primary indication, we envisage severe nosocomial infection ranging from HAP, so bacterial ventilation-associated pneumonia or hospital-acquired pneumonia, complicated urinary tract infection, or bloodstream infection. Mainly caused by gram-negative escape pathogens, which are resistant to the first-line treatment. As clinical indication, the complicated urinary tract infection would be considered as a proof of concept, and HAP-VAP as a second indication to be pursued once the proof of concept obtained. To be able to cover this kind of infection, a new drug needs to cover as a spectrum non-fermenters like Pseudomonas and Acinetobacter and Enterobacteriaceae. The coverage of TPP-associated relevant gram-positive species would allow to cover a full, let's say, optimal preferred TPP. The coverage of only gram-negative pathogen would cover the minimal or base case TPP. Ideally, a new drug should have a new mode of action, so it will be positioned as first in class. And then in terms of efficacy, not inferiority compared to the standard of care, no safety liabilities. And then in terms of dosing and administration for this kind of infection, 7 days, whether or not 10 days should be envisaged. If you could please move to the next slide. This is a kind of flow chart of activities that will support in A to lead phase of a project. I'm showing that because, of course, in vivo pharmacology is critical. However, we have to keep in mind that, of course, the in vitro data are also critical, because at first step, they would somehow support the selection of the best representative hit series or compounds having the best chances of success. And then, of course, based on the in vivo data, you may identify new in vitro data that may be needed to further profile a new series. At the beginning of a to lead, a potential TPP is set up. And then, of course, at the end of the E to lead, ideally, the combination of in vitro and vivo data would support the confirmation that this is indeed the intended TPP that could be achieved. To give a bit more detail, as Erin gave already quite some hints on how the flowchart may look like during hit to lead phase. As quick overview, so in vitro microbiology will concentrate first on the panel of spectrum, potency of the compound, initial de-risking of resistance development, and mode of action, eventually in target validation activities. In terms of ADME and DMPK, at this phase, the project would mainly concentrate on toxicity, protein binding, stability of the compound. In terms of in vivo tolerability and pharmacokinetic data, here maybe the position I put in this flowchart would be a bit different to what Erin would support. Of course, preliminary tolerated data are important, especially because this is often the requirement to be able to test compounds in animal. Pharmacokinetic profile is debatable whether you need to have them before to assess the potential of infected tissue attainment of the compound, or could be used afterwards to profile that more in detail. Based on my experience, sometimes having preliminary pharmacokinetic profile may avoid to test compounds that indeed didn't really have the chance to achieve the infected tissue. And then if you do PK, tissue distribution may also help in directing which would be the disease-related models that are higher chances of success. In terms of in vivo proof of concept, we tend to suggest as initial in vivo efficacy a simple sepsis model, because in this case the compound does not have to cross different tissues, and it would be direct evidence that if you inhibit a specific target in a specific bacterial species, you may indeed achieve in vivo efficacy. Then, of course, the next step would be to show efficacy in a vivo model for several reasons. First of all, because the fact that the compound needs to achieve the muscle as infected tissue, it's an additional layer of difficulty, but would be a different, let's say, a final proof that this is achievable. And last but not least, Phi will indeed be one of the favorite models for the following PK/PD modeling. I'm saying in most of cases because I'm showing a showcase where this is not applied. Overall, at the end of it to lead, the combination of in vivo and in vitro profile will define whether the intended TPP can be addressed during the lead optimization with a lead series identified with a specific characteristic that may eventually be optimized. If you could please move to the next slide. The main goal of lead optimization would be to expand the in vitro to in vivo pharmacology and translatability. And in this case, at the end of the lead optimization, the position would be either you confirm your intended TPP or there are some evidence both in vitro and in vivo that this is not achievable, and eventually the TPP to be addressed with the project can be changed. In terms of in vitro microbiology, the panel of spectrum will be enlarged with a significant larger number of isolates. Time kill curve, but also post-antibiotic effect will be tested against a larger panel of isolates. There will be a full resistant de-risking in terms of frequency of resistance and serial passage. And if your new antibiotic is intended to be given in combination with the standard of care, of course, it would be rather critical to do also combination studies. In terms of ADME and DMPK, PK studies will be performed more in detail, often with the ascending dose and measurement of assessment of PK dose linearity for the compound or for the lead series, and towards the end of lead optimization, also including the metabolite identification in most of the programs. In terms of in vivo model, it will be important to start to identify models which are reproducible and with low variability in terms of group of treatment, because this will be used to identify the driver of efficacy, especially later on, and be used for PK/PD modeling for prediction of the human efficacious dose. These models can be used also to optimize the dosing regimen. So ideally different strains with different susceptibility should be included to cover all the potential susceptibility of the lead series or lead compound itself, and eventually preliminary in vivo resistance assessment can be also included at this stage. And then last but not least, of course, efficacy in a disease relevant model should be assessed to confirm that the compound may achieve the infected target tissue. More complex administration routes can be envisaged, like inhalation or infusion or eventually precision pumps, if this is a route of administration which is envisaged to be used later on in human. And eventually other species than the mouse can be tested for certain models. And for sure, as Erin mentioned, neutropenic mice are the standard model or preferred in early phases to exclude the involve the immune system effect or contribution. However, this is an aspect which is critical to be also taken into consideration in profiling lead candidates. Is at this stage, non-neutropenic models should be also investigated from my point of view. And then, of course, at the end of lead optimization, the in vivo and in vitro data together will confirm if the TPP is achievable by the candidate that will be put through IND studies and later on into the clinics if successful. If you could please move to the next slide. This is a case study where I want to somehow show the journey of a project, showing that the optimization of a drug in IMR space is, of course, a cross-disciplinary approach. The optimization needs the support of is a multi-parametric effort. This project started with target validation, with a lot of in vitro data that were concentrated on defining the potency of the compound, running MIC in a large panel of isolates, including multi-drug resistance strains, the mode of action confirmed by bacterial cytological profiling, and also the risking of the project showing that the lead series had low frequency of resistance. In terms of preparation of the in vivo profiling, time kill curve were performed, because this is one of the parameters that could be taken into account in deciding which dosing will be then required in the in vivo model, eventually combined with the PK exposure and half-life after dosing. And for this program, also combination with the standard of care were done in order to support the drug positioning. The series had the likelihood to pursue two different therapeutic indications. In terms of in vivo profiling, PK were performed, including evaluating different routes of administration, and then the hit were profiled in both sepsis, and after confirmation of efficacy, such a model in thigh model. And as mentioned, the two potential clinical indications were lung infection and urinary tract infection, and the lead series indeed showed good efficacy, very promising efficacy in both models. The project continues nowadays with the goal to identify a clinical candidate. And if you could move, please, to the last slide. This is an example on how in vitro and in vivo profiling of clinical candidate continues beyond clinical candidate nomination and phase 1 in this case. So this slide presents the PK/PD modeling of myrpavadine. In this case, the neutropenic lung infection model was used for modeling for several reasons. First of all, of course, the dose linearity of PK was shown both in thigh and lung. However, in lung, there was a slightly superior efficacy of the compound, most likely because myrpavadine accumulates well in the epithelial lining fluid of the lung. For this reason, this model has been chosen for PK/PD. On the right-hand side, you can see a table which summarizes the in vitro potency of the drug against a panel of 15 Pseudomonas aeruginosa isolates. Myrpavadine is an antibiotic which is specific for Pseudomonas and was intended to be developed for HAP/VAP treatment. As you can see, most of the isolates in this panel are multi-drug resistant, whether not extremely drug resistant, and myrpavadine showed MICs in the range from 0.125 to 1 microgram per ml. Most of the isolates have an MIC of 0.25 micrograms per ml because based on the results of a large surveillance studies, which included about 2,000 Pseudomonas recent clinical isolates, the MIC90 of myrpavadine was 0.25 microgram per ml. This outlined the importance of having a range of susceptibility of the strains used for PK/PD modeling, able to cover the MIC90. And one isolate with which was less susceptible, with an MIC of 1 microgram per ml, was included to be sure that higher MICs would be covered with what will be at the end the predicted human efficacious dose. And the bottom of the slide shows the in vivo dose response of myrpavadine against 3 Pseudomonas isolates. Each symbol in the graph shows the therapy response in one lung lobe, showing that this was significant study where a large number of animals were included. The next step was then to do the fractionation of the total daily dose to be able to identify and confirm what is the driver of the efficacy. And later on, the human PK was predicted and compared to what was the human PK, which was indeed available at the time the study was completed, and confirm what would be the dose which is expected to be efficacious in phase 2 clinical trial. In general, there is also the possibility, but this is an option, to also build up an in vitro PK/PD data model. An example would be to use the hollow fiber system. The hollow fiber system can be used to mimic the exposure to the bacteria based on the human PK/PD, or human PK, and that would confirm the in vitro capability of the drug to kill different isolates with different susceptibility and different phenotype based on doses that would mimic the human exposure. Or also to determine what would be the likelihood of resistance development based on treatment sub-exposure for several days. And with this slide, I've concluded my presentation. Thanks. And thanks, Frederik, for changing the slides on my behalf.
No problem at all. No, thanks as well, Francesca. So let me see. I don't think I see anything in the chat. Tolga, did I miss anything? Because I couldn't see it when the slides are shared. I think we can then.
No questions in the chat.
All right, good. Maybe just a quick one that I fully understand. So the TPP is a dynamic document. So I think that's what you also explained. And so you start with a potential TPP, and then an intended one, and then a confirmed TPP. So basically, how do you do this in practice? So you update these TPPs all the different stages or during the duration of the project, or how much flexibility do you have to change the TPPs? I know it was also part of the previous webinar, but maybe some people did not attend. So
I would say that is a kind of bilateral process. On one hand, the TPP gives you a guidance and a kind of checklist whether you may achieve, or your product has fulfilled the criteria that would be needed to achieve a certain TPP. The other way around, if you realize that your lead series or lead candidate is not likely to achieve the TPP or has not criteria that are compatible with the TPP, it would be good to revise it. And this is indeed a very dynamic process. Mirpavad is one of example of this flexibility, with harsh flexibility. However, as you may know, the compound went to up to phase 3 for HAP-VAP, the clinical development, and it was stopped. Was in HAP-VAP as IV treatment, and the drug has been repositioned as inhaled therapy for the treatment of cystic fibrosis in CF patients, and that was a full change of TPP. That was feasible because at that time there were a lot of human data in terms of PK, a lot of human data in terms of efficacy. So to be able to reposition the drug as inhaled therapy, let's say small bridge studies were sufficient.
Thanks, Francesca.
There is a question in the chat. Frederik, did you see it?
Yeah, so from Elizabeth. Elizabeth, do you want to tell your question? Otherwise I can read it, but otherwise you can go ahead. Elizabeth?
Sorry, can you hear me?
Yeah.
Okay. So I wanted to ask, when you want to determine your MIC50 or MIC90, ideally, how many strains should you use?
It will strongly depend on which phase of the project you are. I would say at the very beginning, at the A to lead, maybe something like 20 to 40 strains may be sufficient. It will also depend on the spectrum of your compound. Of course, if your compound is narrow spectrum, it will be easier to have MICs on 20, 30 isolates. If the compound has broad spectrum, this panel could be the same size, but with lower number of isolates per species. If the project then progresses, the more you have, of course, the more solid would be the data package and the prediction that you can perform based on the data. You can go from anything between, let's say, 40 in an early phase to clinical candidate, when, of course, you have to do surveillance studies and measure the MIC50, MIC90 on maybe 2,000, 3,000 different isolates, and especially that needs to be recent isolates.
Yeah. Thanks, Francesca, and thanks, Elizabeth, for the question. Very good. All right, Francesca, if you can also stay for the panel discussion at the end. We have some more questions for you, but we'll first gonna go to the third speaker. Jennifer, if you can also introduce yourself first, I think that's always good, and I will then do your slides as promised. Huh?
Wonderful. So I'll introduce myself while you pull up the slides. Can you hear me okay?
Yeah, okay.
Okay, great. Hi, everyone. Good morning or good afternoon, depending on where you are. I'm Jen Hoover, and I'm from GSK. And I have been conducting animal infection models there at GSK for almost 30 years. And I'm really happy to be here today to talk about how we incorporate in vivo models into our antibacterial drug discovery process. I will be focused more on small molecule antibiotics. There are some unique challenges for non-traditional approaches that I'm not going to get into in the presentation today, although perhaps in the panel discussion we could delve into those weeds if anyone is interested to do so. And Frederik, I don't see any slides yet, so.
Yeah, I was just waiting until you have your introduction.
Oh, okay.
Let me see.
Well, my talk is probably 20 minutes long, so I'm going to kind of launch into it while you're sorting out the slides, if that's okay.
No worries. Can you see the slides? Is it okay?
Yep, that's great. Thank you. So go ahead to slide number 2 when you're ready. So I unfortunately couldn't make the TPP talk, so I'm not exactly sure what all has been discussed, but I thought it would be good to at least start by going over the 3 preclinical phases that we generally have at GSK. So we start off in the target identification and confirmation stage, and this, of course, is the earliest phase. You probably won't have any of your own compounds at this stage, but there may be some reference compounds or tool compounds that you can use for testing. We then move into lead discovery, which is the time when we start to run high throughput screens to actually find novel compounds that can modulate the target that we've chosen in the first phase. And from there, we move into lead optimization. And this is where we really work to improve the compounds and the chemical series in order to find that one special compound that we think actually has what it takes to become a real drug. And also for my talk today, I'm not going to be talking about the additional work that we'll do during the clinical development stage. I'm really going to focus on what we do during these first 3 preclinical phases in terms of generating in vivo data. Next slide. So when you're thinking about incorporating in vivo models into your project, it's going to depend on the goals of your project, every study, the goal of every individual study, as well as what you're trying to achieve at a particular given time. So how you design and conduct the studies is, of course, going to be dependent on what the goal actually is at that time. So when you're very early on, like target identification, for example, then you may just be looking for a proof of principle. You're looking for a reason to believe, is this a good target? And you may be interested to explore a bit more about the mechanism of action. But as you move into the lead discovery and lead optimization phase, now you really need to start thinking about the compounds as drugs. So do they have developability qualities that you're going to need? You're going to start to compare different series and different compounds within those series. And then ultimately, you need to confirm that your candidate actually meets the criteria for the TPP that you've laid out. No matter which phase you're in, there's some important ethical responsibilities that I just want to remind everyone of, because we are talking about using animals in scientific research. So first of all, you should always use non-animal alternatives whenever possible. You shouldn't use your animal models for high throughput screening. That's where your in vitro assays come in. And also when you're thinking about testing in vivo, carefully think about when is the right time to test and what is the right thing to test. Never try to understand how your in vitro assay data and your in vivo data correlates, because you can actually use this to potentially some of the number of in vivo studies that you might run, or at least better use in vitro data to design better in vivo experiments. Next slide. So this is a snapshot of some of the common goals that you may have at each phase. In target confirmation, as I mentioned, you probably won't have any of your own compounds yet, and you may just be looking for, again, a reason to believe at this point. So you may be able to test benchmark or reference compounds, but one important note here is if you're going for a novel target, there may not be any compounds that you can test. So this may not always be feasible to run in vivo studies at this. You may also run into an issue where even if there is a reference compound, you may not be able to obtain it in enough supply to put in vivo, or it may not have adequate solubility or other characteristics that allow you to even put it into an animal in the first place. As we move into lead discovery, now we're really talking about looking at compounds that you have, your novel compounds. Here's where you can begin to generate some in vivo data, generate in vitro data, and look for those correlations, as I actually mentioned. You'll start to think about comparing different compounds, different series of compounds, and you're going to want to start thinking more about, can these compounds actually behave like a real drug? And it just gets harder as you move on to the lead optimization stage. At this point, of course, you really want to start asking your compounds to do a lot more. They need to have real drug developability properties that are consistent with a real drug that can be successful in the clinic. You're going to need to confirm that your compound actually can meet all the criteria of your TPP. Next slide. So when we start to move to the question of how you decide which in vivo model to use, I wanted to start off with kind of a list of some do's and don'ts. So first of all, do use in vivo models and bacterial strains that are aligned with your TPP. Second, do try to simulate the human disease, but don't expect a perfect animal model, and I'll come on to this in a minute with one of my don'ts. The third do I have is do rely on validated models where possible, and Erin touched on this as well. Don't reinvent the wheel here. If there is a validated model that's already been shown to translate successfully to the clinic, then you're just going to be 10 steps ahead if you use one of those models instead of embarking on something brand new. In terms of don'ts, please remember that mice are not furry test tubes, so don't inject your bugs and your drugs in the same place at the same time. I also encourage you to think very carefully about the models you choose and don't use self-resolving infection models. And I'm going to talk about one of these in particular on my next slide. And I suspect it might be the model that Erin alluded to in terms of UTI. My third don't is don't try to copy the human disease precisely. And the reason for this is it might be impossible to develop an animal model that will mimic exactly the pathogenesis and the course of disease in a human. So do get a good proximity of the human disease in your animal model, but don't let pursuit of a perfect animal model stop you from actually testing your compounds in a good animal model. Next slide. We're really lucky in our field that we have a number of choices when it comes to infection models. So I guess this is a pro and a con because it means you have to make choices here. So I wanted to go through each of these indications and talk a little bit about different models. It is really important before you start on a project that you understand the precedents that have been set. And as I mentioned before about using validated models, this goes really along with that same point. So don't reinvent the wheel. If someone has already used an animal model and that's been shown to successfully translate to the clinic, that's a good place for you to start. So I'm going to run through these one at a time. First of all, for pneumonia, you can do some fancy things by doing, a thigh infection model or another model, and then looking at, using modeling to compare plasma levels to ELF and so forth and so on. I just think we're beyond that in 2024, and there are some quite good lung infection models. And as Erin mentioned, we are working to validate one as part of Combine. So really, I would encourage you, if pneumonia is your indication, use a lung infection model. For complicated UTI, the old standby is the neutropenic thigh infection model, and this is a fine model to use. It's been shown to translate, and so I think that's a good choice. Another model I wanted to mention here is a pyelonephritis model. So we have started running this model at GSK, and I've seen one or two other labs run this model where you directly inject bacteria into the kidneys, and it develops a very robust pyelonephritis infection. It's not self-resolving. And we're actually currently using this model to do PK/PD studies for one of our phase 3 assets. So I'll tell you how well that works later. For UTI, this is an interesting situation where most people are tempted to use the mouse cystitis model. However, there's a lot of caveats to this model. There's not a lot of strains that take in the model, and it's generally self-resolving. So I consider the cystitis model to be quite permissive. And I think compounds will get through this model when they shouldn't. They probably will work in this model, but that doesn't mean they're going to work in the clinic. The only exception to this, I would say, is if you have a mechanism of action that is local, acting in the bladder, then you don't really have a choice and you would have to use this model. But otherwise, I would steer clear of it because you can be misled. What I would suggest instead sounds strange, but bear with me. So you can use a thigh infection model if your goal is really to characterize the PK/PD of your compound against a particular bacteria. So for example, for UTI, you would typically be looking at E. coli, Klebsiella, maybe Proteus. You can do the PK/PD in the thigh infection model, and then you can use the plasma levels that are associated with efficacy in that thigh model and correlate that with urine levels in the human bladder. This sounds a little strange, but this is actually what we've done for our compound gepotidacin. We used PKD from a thigh infection model, we used humanized dosing from a pyelonephritis model, and we used one compartment chemostat models and a hollow fiber model. All 4 of those went into our dose prediction for gepotidacin. And as you may be aware, we have currently reported successful outcomes from 2 phase 3 trials for uncomplicated UTI. So this was a really nice approach for us that was successful. For intra-abdominal infections, there's the old standby again, the thigh model, and that's the one I would recommend. There are some sepsis models, but I'm personally not very keen on most of the sepsis models. So again, following what's been done before, I would just use the thigh infection model. For skin infections, we again can use the thigh infection model, but there are some abscess and wound models that I believe have also been used to successfully translate to the clinic. No matter which of these models you use, I highly encourage you to validate the models and the bacterial strains that you're going to use within your own lab. And also, if you're working with a partner, BARDA, CARB-X, or another organization, I would always gain stakeholder agreement on which of these models you're going to use before you start conducting studies. Because if you get down the road and you come and you present some data and they don't agree with the model you've chosen, then you're going to have to backtrack, and that will waste time, resources, and even more importantly, animals. So always get agreement before you embark on the project if you're working with a partner. Next slide. So this slide, there's kind of a lot of information on this slide, and this is really getting into some specific suggestions for how to design the studies at each of the different phases. If you are very early on and you're just looking for a proof of principle with a reference compound, basically this is your reason to believe type of study, then in my opinion, I think you could use pretty much any organism. You could start with just one strain that you know works well in the model. I wouldn't worry too much about the MIC of that reference compound. I wouldn't worry about the dose regimen that it takes. I would just probably start with a high tolerated dose just to show that I could see something positive that tells me that I'm directionally, going the right way. In terms of controls, I generally agree with what Erin said. You should always have a positive control in your studies. I didn't put that here mostly because when you're very early on, there may not be a positive control, but you should definitely have a growth control in these experiments. And this would be your vehicle treated animals, because you want to know your model is actually performing, that bacteria are growing, because if they're not, again, you don't want to be misled thinking your compound has actually caused a reduction in bacteria when it's really just the model is not running very well. As you move on into lead discovery and lead optimization phase, then you're going to start wanting to compare different series, compare different compounds, start to really understand if these compounds are going to have what it takes. So when you get to this point, I would start to look at least one strain of 2 to 3 of the relevant pathogens for your TPP. I would not just test at the lowest value. I would test at least at the mid-range, potentially even starting to think about looking at your strains that have your MIC 90. If you are comparing 2 compounds, I would try to choose a strain that has, a similar MIC for both of those compounds. Otherwise, you're going to be giving one of them an unfair advantage. At this point, I also like to start to understand how the compound dose respond. And so I would like to look at, about 3 doses of each compound. Again, early on, I wouldn't worry too much about the dosing regimen, but do keep an eye on this, and I'll come on to that a little bit more when I get to the final example here. I would also include your vehicle-treated animals as your growth control, and at this point, I would include a positive control. Now, of course, if you're looking at XDR strains, there may not be a positive control, but whenever there is one, I would always include it. And one of the things that we do at GSK is even if we can't find a marketed drug that can be used as a positive control, we'll have novel compounds that maybe can't make it through for some other reason, but they can serve as their own positive controls in some of these experiments. One last point here, if you are comparing one compound to another, I would always encourage you to test those side by side in the same experiment. Study to study variability is real, and we have to acknowledge that. And if you're comparing the efficacy of one compound to another compound in 2 different events, unless it's very black and white, one works and one totally doesn't work. If it's kind of, a bit more gray, then you can be misled if you're looking at from one experiment to another. And so I would encourage you to test these compounds side by side in the same study. Lastly, when we get towards choosing that final candidate compound that we want to take forward, then you really have to get serious. So this is the point where you're going to need to confirm that your leads can meet the criteria. And I would do studies with at least 2 strains of each of the relevant pathogens for your TPP. This doesn't mean you have to test against every pathogen in your TPP, but choose the key ones. So for example, if you want to go for Enterobacteriales, I would at least do E. coli and Klebsiella at a minimum. You want to start to test strains across your MIC distribution, although I would prioritize strains at the upper end versus the lower end, because again, if you don't show that your strain can work against the MIC 90 at this point, you could be in for a nasty surprise later. I would also continue to want to understand how the compounds dose respond. So I would do, at least 3 dose levels of my compound. For the dosing regimen, again, I'm going to echo what Erin said. I would, for rare cases, I would be okay having to continue to dose compound every 2 or 3 hours at this point. You have to start thinking, does this compound have what it takes to be a real drug? So generally, you're looking at, less than or equal to 4 times a day. There are exceptions to this. So for example, we know there's, a number of carbapenems that have super high clearance in mice. They're better in people. And some of those, you may have to dose every 2 or 3 hours, but yet we can still also, I wouldn't do, again, echoing Erin, I wouldn't do PK. Maybe I would do PK before this, but I wouldn't focus on it too much. But at this stage, I would want to start to understand my exposure response relationships. So I would generate PK data. And importantly, we have found in our hands, at least, that it is always best to do PK in your own studies. So using infected animals, running the model exactly the same way as you would for an efficacy study is what you want to do, and then take your samples and have them analyzed and generate the PK in that way. That way you can directly correlate PK that you have in that animal model with the efficacy that you're getting, and it'll give you an early read on your human dose prediction, and also help you start to understand the PK/PD characteristics of the compound. Next slide. Oh, actually, before I move on to the next, I just want to say, so my last several slides, and actually even my next slide, I don't want you to take everything I'm saying as the gospel truth. This is what I recommend or suggest from my experience. So, always be sensible. But I'll come on to this as actually as I get to my last slide. So don't write all this down as if it's completely and utterly set in stone. Along the same lines as my last slide, where we get a little more rigorous as we move through the phases in terms of how we design the study, how we interpret the data from the study can also, or potentially should become more rigorous as we go along as well. So when you're very, very early on and you're looking for a reason to believe, you can potentially ask a little bit less of the compound, especially if it's just a reference compound or a tool compound. So in that case, it might be okay to say, okay, is the response I see statistically significant reduction versus my growth control? That can give you a reason to believe. But as you move through the phases and you're starting to look at leads, and especially as you start to assess whether or not your selected compound is actually good enough, you need to get more rigorous in terms of how you interpret the data. By the time you're ready to choose your candidate, you really need to be asking these compounds to give you at least a log reduction, which is statistically significant from baseline. I didn't talk about this before, but always include baseline controls. So this is a group of animals that you take at the time you start dosing, which is generally an hour or 2 after infection. You want to know what the CFU is in those animals at that time that you start dosing, and that becomes the yardstick by which you measure efficacy. So we know that real drugs, successful antibiotics in the clinic, can give us this 1 log reduction versus baseline controls. And if you want your drug to be able to compete, you need to ask it to do the same thing. Next slide. So as I mentioned before, these are suggestions. It's not a Bible. For better or worse, there really is no single right way to do this. At all stages, I just encourage you to be sensible. You don't want to be too tough too early, or you could actually, kill a project before it really has a chance. But on the flip side, don't be too easy too late in the process, or else you're going to end up with a candidate that really can't do what it needs to do. So I'll end with these 4 bullets that I have here, which is to engage experts along the way. Talk to people, show them some data, get some feedback. This can be a really important reality check to make sure that you're on the right track. I mentioned this before, but I will emphasize it again, involve your stakeholders. So if you're working with another group, like BARDA CARB-X or another group, involve them in the process. They are probably financially invested and they are probably decision makers for you. So you want to be sure that they agree with what you're doing throughout the process. It is very easy to get emotionally attached to our projects. And sometimes our compounds become like our children. And so I want to encourage you to listen to the data and try to take your heart out of it as you're making decisions about whether things are good enough or not. And following on from that is to always be a truth seeker. The harsh and unfortunate reality is that most compounds are not good enough. And sometimes it can be the hardest thing to do to say no to a compound or even to stop a project altogether. But sometimes that is the right thing to do if it's really not working out the way it needs to. So design your in vivo testing cascades in a smart way, be tough about it, but be fair about it. And then when you finally hit upon that rare gem of an asset that can make it through all the tough tests you've thrown at it, then your victory is going to be a very sweet one indeed. And thank you, and I'm happy to take questions.
There is a question for all three speakers, and maybe Jen could start on it by commenting on efforts to standardize animal models.
Yes, so I mean, the only one I am aware of is our efforts in Combine, where we are working to standardize a lung infection model. And thank you, Erin. Sorry, I see the comments popping up here. Thank you, Karina. Yeah, so that's underway, and I think we're making really great progress on that. And we're very fortunate to have some partners that have also joined in with Combine. And I want to just say a huge thank you to Erin and CARB-X for their efforts in this. And you know, if anyone wants to contribute to our effort here, we are always happy to take willing partners. We can't fund you, but we are happy to take partners to help us generate data for this effort. And I don't know if anyone else is aware of other standardization efforts, but yeah, happy to hear from you.
Thanks, Jennifer. I hope you learned a lot as well on the importance of the in vivo. I think it's nicely linked to the previous webinar. And we will have a next webinar as well, probably in the fall. So we're going to continue to share success stories, also some failed stories as well. So I hope you enjoyed it. And I see already some people that shared their enthusiasm on the chat for thanking you. So thanks a lot, everybody. And thanks for attending the webinar. Thanks.
Thank you.
Everyone. Bye-bye.
Bye.
Bye.